Welcome back. The 1953 landmark paper by Watson and Crick was published with just two figures that were models of DNA structure and its implications. The paper was published at the same time as a few other papers by Wilkins, Franklin, and others showing actual data that backed up the Watson-Crick model. Figure 1 of the Watson and Crick paper showed the classic double helix we all know. If we look a little closer, we see A's on one strand across from T's on the other strand, and similarly, C's on one strand across from G's on the other strand. There in the DNA structure is the basis of Chargaff's rules number 1 and number 2. The amount of A is equal to the amount of T. The amount of G is equal to the amount of C. And the amount of purine nucleotides is equal to the amount of pyrimidine nucleotides. Watson and Crick came to call this complementarity. The two strands in a DNA helix are complementary to each other. The incredible power of this finding means that if I know one DNA strand, I automatically know its complementary strand. Understand complementarity, and you know how PCR, or polymerase chain reaction, works. Know complementarity, and you can see how scientists can construct single-stranded DNA probes to detect specific pathogens in a blood sample. But look again a little closer at just one strand of the DNA double helix. The sequence on that one strand changes as we move along it. It can potentially be anything. This means that the sequence of DNA can be, and often is, different between different organisms. Chargaff's rule number 3 comes alive. If that is true between organisms, it can also be true between different parts of the same DNA molecule. Sequences over the shorter range differ, and that can allow each gene to be unique. Unique genes can have unique functions. The simplicity of DNA makes us underestimate its real power. Figure 2 in the 1953 Watson and Crick paper shows a DNA double helix with part of it unwound into single strands. Along sections of those single strands, we see new complementary strands being made. In the text of their paper, Watson and Crick did not explain this figure in detail, but rather stated matter-of-factly that their model of DNA structure made it obvious to how DNA could be precisely replicated. Their figure 2 is quite obvious. Each strand of the old double helix is used as a template for the making of a new complementary strand. In other words, each daughter cell carries one original DNA strand from its mother cell, along with a new complementary strand. If you think about your own body developing from a fertilized egg, there are now less than 100 cells in your body that actually have a DNA strand that was present in the fertilized egg. All the remaining DNA strands in your body have been made using complementarity. The Watson and Crick model changed the world forever because of the implications of DNA structure on how it is replicated, how it changes, and how it can code for proteins. However, as we will see in future episodes, a model that makes predictions still must be tested. Thanks for listening.